Okay, I think we can slowly get going. I'm going to start with some announcement, and then we'll present to you the three speakers of the day. This seminar and the next one on the 30th of January are going to be special seminars to highlight the CLIMACT Starting Grant grantees from 2021 that are just finishing their projects. So today we will hear from three of those researchers and projects, and then in 2 weeks from now, on the 30th, the four remaining researchers and projects will be presented to you. So, as I said, today we will have three of these CLIMACT Starting Grant project from 2021 highlighted. The first one will be by Dr. Anna Carratala Ripolles from ENAC at EPFL. Anna is a research scientist in the Environmental Chemistry Laboratory at EPFL and an expert in environmental microbiology and virology. Her latest research involves the characterization of the diversity of microbial communities of the Rhône watershed, including alpine and peri-alpine lakes across space and time, and the adaptation of microbial communities to environmental changes. She is also interested in the development of novel nature-based biotechnological applications. The title of her talk for today, 2000 Lakes, Alpine Research and Citizen Science for the Microbial Conservation of High Mountain Lakes. Hello. Thank you, Anna, for being with us.
Hello, thanks a lot for the introduction. I'm going to share my screen. Great. So as you introduced, I'm going to speak about the project that we have been developing for the last year, thanks to CLIMACT Starting Grant. And in this project, we've been working with very beautiful ecosystems that we have here in Switzerland, Alpine Lakes, which are located above 2,000 meters of altitude. And my first slide is about why do we care about these environments? They are far away in the mountains, really remote, but they produce a lot of services for human populations and for animals and other species in the alpine environment. Like, for example, they provide a source of water and food for many organisms. They also contribute to regulating climate, for example. They also support many species as an habitat, and they support the development of food web dynamics. They also contribute to soil formation, nutrient retention and cycling. And of course, they are important for human populations in the sense of, education, spiritual values, and as a recreation and tourism spot. So they have a role in our economy, particularly after the snow era, right? However, these lakes are sensitive to climate change, as you can imagine. They are exposed to an increase in their surface temperatures, and of course they experience less ice-covered periods with these mild winters that we are experiencing. There is also, due to an increase in temperature, an increase in the evaporation. So sometimes these lakes, which are typically not very large or not very deep, like the one in the pictures, they can even evaporate fully, which is something that happens already like naturally, but more and more often. And this has an impact for all the biodiversity that it's connected to these ecosystems. There's also less frequent mixing events, so this changes the ecology of the ecosystems. Altogether, this has an impact in the ecological functioning of this ecosystem, which so far cannot be fully anticipated. So this takes me to explain some of the research questions that we had in the project and in my research. So I'm interested in understanding a little bit better which is the diversity of bacteria, of microorganisms that are depending on these ecosystems, and what do they do? How do they use the space and the water column? How do they respond to environmental changes? How do their responses to climate change can actually impact the functioning of the ecosystem? and what is a healthy or an unhealthy ecosystem. But the ultimate question is, should we actually protect microbial diversity, which is the basis for the functioning of these ecosystems, as we do for other threatened species? So in this project in particular, we studied a total of 60 lakes across the Swiss Alps, and we were focusing on collecting water and biofilms in these environments to focus on the bacteria communities inhabiting these lakes, and also with a particular focus on toxic cyanobacteria, which is a type of bacteria that are known to be favored on occasions by climate change and warming temperatures. We have built a collection of bacteria, more than 220 isolates so far. And when we collected the samples, essentially what we did, for you to understand if you're not in the field, we collected water or biofilms, and we typically filtered the bacteria to retain them on a surface, on a filter, which we could then later either isolate or process through molecular biology techniques to extract their nucleic acids that we later characterize by metagenomics and by different ecological analysis to understand community composition, to estimate their diversity indices, or to develop other correlation analysis which allow us to connect the environmental measures that we characterize with the community composition. I'm going to show just few results. I'm not going to go into a lot of detail. I just want to give an overview of what we've done in the project. But here what I'm showing is a graph representing the different diversity indices that we have calculated for different lakes, organized by the region. And we can compare that with the diversity of bacteria that we have observed in Lake Geneva. So the graph below corresponds to the data that we have collected in Lake Geneva in other projects. So this is just as a reference. And we see that the diversity in this lake can span from few hundreds to up to 1,000 of this index, which is calculated taking into account how many bacteria there is in the ecosystem, with a small statistical correction for the minor variance. The most important point of this graph is to show you that actually the diversity in these alpine lakes is in general higher than the one that we have observed in Lake Geneva. And this is important because these lakes can actually represent hotspots of bacteria diversity. This was something that was surprising to us, as we thought that maybe, you know, the mild conditions experienced in Lake Geneva would favor more diversity, but that's the opposite. There is more bacteria diversity in alpine lakes compared to temperate lakes. Also, without going into detail, what I want to show you here is that we have observed that these alpine lakes are very different from one another. Even lakes that are very close, like for example, I indicate two examples here, like Gringisee and Grundsee, which are two lakes that are in the Zermatt region, and PA1, PA2, PA3. These are three small lakes that are located in the Aletsch Glacier region. These bars here there's no need to go into detail, but imagine that these bars are kind of like code bars characterizing the composition of each of these lakes. And you can see that even lakes that are very close by have very particular and distinct bacteria composition. What this means is that even close lakes that you might expect follow same environmental conditions are actually very different. So very localized characteristics will have an impact in the community composition and lake functioning. So we know now that lakes are very high in bacteria diversity, and even close lakes like these are PA1, PA2, and PA3, as I just mentioned. You can see that really they are few meters away from each other. They are very unique ecosystems. And which are the local, you might ask, which are the local conditions that, make this lake special? So first of all, we've seen that some of these lakes, even located very nearby, have different temperatures. So sun exposition and depth can have an impact on the temperature of the water, but also the bedrock. Jasmine Berg, who is a collaborator in the project, has characterized the biogeochemical context of these lakes. And we see that different elements, like, for example, iron or phosphorus and sulfates, can have an important role in determining which particular species can thrive in one ecosystem or the other. So as I mentioned before, we have also focused on monitoring the distribution of toxic cyanobacteria in these lakes. These are just few pictures of different cyanobacteria mats that we observed in the lakes that we have sampled. And these species I mentioned before, but they are important because climate change is known to favor them. They thrive in conditions where temperatures are higher, and they are not limited, or not as limited as other bacteria, by poor nutrient conditions. Therefore, they are known to be able to thrive. And we wanted to know if these lakes have these potentially toxic cyanobacteria. For that, we have used molecular assays that allow us to identify the presence of one fragment of gene that codes for the toxin that these toxic cyanobacteria can produce, one of the toxins, which is called the microcystin. And as I'm showing you here, you can see that some of the samples, and there is no labels here, and this is done in purpose because we don't want to alarm anybody by the results we're showing here. We are conducting still some research to understand a little bit better which are the species, and further analysis to make sure that this is really what it looks. But according to these results, we can say that we have found some toxic cyanobacteria in some of the lakes that we have sampled, and these results were confirmed by also collecting samples of the biofilms itself. So they are there, but we are still conducting some more research. But this project was not only a research project, as I mentioned in the title of the talk. It was also a project where we were aiming to connect with people and really have or create a bridge to connect with the society. And for that, we have built a network of stakeholders. We have also developed different surveys for students and to understand what their interests would be in a project like the one that we have been developing. We have also produced other surveys for the general public about their connection and their relationship with Alpine Lakes. This is a QR code that could take you to this survey. If you're interested in actually answering our questions, it would be super helpful since we are continuing this research. We have also developed pilot sampling campaigns, inviting citizens to come sampling with us. You can see some of the events that we organized in these pictures. We have also done a little bit of communication efforts and outreach efforts. We have an Instagram account that you can actually follow. Now it's a little bit silent, but there is some information about the work we have done. And we have also built a website that it's not yet public, but it will be soon, in which we have collected information about all these lakes that we have studied and others. And the goal of this website is to actually make the digital existence of these lakes grow, lakes that are actually threatened by climate change. So as future directions of this project, I would say that now it's a time to really reach out to this network of stakeholders that we have built, share our results with them and understand what is their interest also with the project to actually see how can we serve from now on. So the project, the project as it was, it's finished, but the effort that we started, it's going to continue. We have established a partnership with a natural park in the Vallée du Trient to continue the monitoring that we already started within the boundaries of the park. And we imagine this project as an initiative. We want to make it grow, and we imagine it as an initiative that can host other projects and really build a wider and stronger effort to act and understand the consequences of climate change in these sensitive environments, and hopefully develop some actions to mitigate or to protect these environments and the microorganisms that live in them. And I mentioned before that we have this collection of isolates, so we want to really establish it, make it public through a website or through some means. This is something that we are still developing, but we want to make it public, and we make these isolates available for education and research, and to make the life of other researchers easier. And with this, I hope it was not too fast. I didn't control my time, but maybe it's fine. And with this, I would like to thank everybody involved in the project. There was a bunch of people, different scientific collaborators, the co-PIs of the project, and many students that participated. By order, here we have Daniel Alvarez and Yuan Huilin, Jessica Oboe, Paul Segan, Lisa Morales. They did a lot of work to make this project successful. And I also want to thank all the people who participated in the game, and they were super open to share their questions and their concerns and their interest. It was a super nice project to do. I was so honored to be part of it, and thank you, CLIMACT, for funding it. Thanks a lot.
Perfect. So let's move on. Thanks again, Anna, for this great talk. We're going to move on now to Professor Bogdan Draganski. Professor Draganski is consultant neurologist at the Department of Clinical Neuroscience at the CHUV. He is the director of the Neuroimaging Laboratory and of the departmental MRI platform. After qualifying in clinical neurology in Germany, he spent time working on computational anatomy research in neurodegenerative and movement disorders at the Institute of Neurology, UCL London in UK, followed by research at the Max Planck Institute for Human Cognitive and Brain Sciences at Leipzig, Germany. His current work is focused on understanding the principles of brain's health span to develop preventive strategies and interventions in the preclinical phase of age-associated brain disorders. His title talk is Climate Change, Air Pollution, Brain and Behavior. So this time around, I got it right. Thank you for being with us, Bogdan.
Well, thank you very much for having me on this panel. And I think particularly my big thank you goes to Charmy, Isabelle, and to Nicolas for being so proactive and really supporting such a great initiative, because I know the funding is important from the UNIL and from the EPFL, but the people behind the whole social media also buzz, this is also very important. Okay, let's not spend any more time in chit chat. My name is Bogdan Draganski. I'm a clinical neuroscientist, and I'll take you on the short journey, now sandwiched between rocks and lakes, on what we did for the climate project regarding climate change, air pollution, brain, and behavior. You ask yourself why, for God's sake, clinical neuroscientists will be interested in climate change, etc. Beyond, of course, some common citizen and social person motivation, of course, my motivation was slightly different. Knowing that in Europe, besides cardiovascular diseases and cancer, brain disorders are on third place in terms of morbidity and mortality. Of course, for me as a neurologist, it's interesting to get an overview of what's happening, of course, in a broader sense in research. And being for so many years in clinical research, I started thinking that we are really in this lamppost phenomena that we are looking for the lost keys under just the lamppost where is light. And we barely look actually at clinical problems outside of the hospitals. You see, we funnel all these individuals that come through different health systems and so on, through the health system, and this is the basis of our research. Epidemiological studies are difficult, and this is my second motivation, being interested also in factors that are associated with neurodegeneration in old age. You know, there are these Lancet commissions on different topics, and this is the report of the Lancet Commission on lifetime factors contributing to 40% of explaining dementia. And they added 3 additional factors in 2020. And one of those, as you see, this is the role of air pollution. That means there was enough evidence, this is really a meta-analytical approach, that also air pollution plays a significant role in the way how the brain is aging, and particularly on our cognitive abilities. I'll give you just some examples. There are enough reviews published on the topic. You see on the left-hand side, the different factors that we are measuring in terms of air pollution. They are really covering a lot of what we call neurological and psychiatric disorders. Again, more and more, there is accumulating evidence that there is a clear causal relationships between the two. And the recent study that I am showing here in brief on the right-hand side was also, for me, a little bit of shake-up. Overpowered, done on 3 million individuals in Australia. They showed that just a temperature rise by 1.5 degrees is leading to a 12% surge in hospital admissions on patients with cognitive problems. And of course, this is paralleled also by enormous increase in scientific publications on the topic. You see, particularly since 2005, let's say 2010, we're really witnessing a lot of work, unfortunately, mostly on the impacts of climate change on health in Europe, also across the world, and less so on mitigation and adaptation. This is also something to keep in mind. Well, I promise to be very quick with my overall slides, and I'll switch here to actually the research that we did together with the person that I saw also here among the listeners. Hello, Stefan. This is my partner in crime that actually brought my thinking about how actually we can study brain and behavior outside of the clinical environment, but in a much more ecological sense, in both senses, also outside the interaction with nature. Stefan was so patient to explain me also some of the basic features that he's measuring in terms of, for example, greenness of the open space, etc. And we were fortunate that in Lausanne, since 2003, there is a large-scale longitudinal study called Cohort Lausanne, COLAUS and CyCOLAUS, with the initial idea to bring together cardiovascular risk factors with risk factors for mental disorders. And as every successful study, it attracted a lot of additional ideas. There is this so-called HypnoLAUS study. That means 2,000 individuals were really deeply phenotyped in terms of how they sleep. There is ActiLAUS, 2 weeks of continuous measurement of activity. GeoLAUS, where Stefan is very implicated, and so on and so on. So this is a cohort that started in 2003 and was representative for the population of Lausanne at that time, and it included roughly 6,700 individuals from the community dwelling population, aged 35 to 65 at the time. And of course, you see this is the typical case where we see that younger individuals are much more suffering from mental disorders, the older ones, cardiovascular disorders. which of course related either be through the cardiovascular side or neurodegenerative side, to low cognitive scores. Of course, also this generation suffers the cliches of women being with a lower socioeconomic status, and so on. From this population, a few thousand individuals, we started 2013 actually to invite all individuals that are motivated to pass through a magnetic resonance imaging exam, where we are collecting data about the brain anatomy and function, a very deep phenotype. We have more than 10 features per each pixel in the brain in three-dimensional space. And of course, we have exhaustive access to a wide range of variables here. Maybe it's too small for you, but this colorful Manhattan plot is just going over the simple correlations between brain structure, cardiovascular risk factors here in green, the strongest one, if you see, how much they surpass the Bonferroni level of correction for multiple comparisons. But we have also inflammatory markers, lifestyle, mood, different measures, and so on and so on. And the first thing that we actually tried to do was really in a very agnostic way to look across different factors that Stefan and his team were capable to objectively measure, to see actually where, in which domain, is there any association between the greenness of area here in the Lausanne city, estimates which are linked to individuals' place of living, of daylight. Then there was a very clever index of walkability, that means access to public transport. Nocturnal and diurnal noise levels were measured also in the housing of the individuals. And of course, we have the usual estimates of air pollution. Here on the y-axis, you see we adopted a certain threshold of eta square of 0.06 to be very conservative about the link between these environmental measures and different measures of mood and cognitive abilities, which are just administered as a test battery. STAI stands for State and Trait Anxiety Index. GAF is global activity of functioning, which is a good measure of the level of mood. Let's say low GAF, it's a scale between 0 and 100. If I'm 50 or 40, I'm very depressed. If I'm 100, I'm perfectly fine. And the mini mental state exam is going across different cognitive functions, is a measure of cognitive ability. And you see already we have certain indication already in this Manhattan plot that the index of greenness is somehow related to the measurements of anxiety that we have. We have also some hits on air pollution, and this is what actually we wanted then to examine or investigate in detail. And here these beautiful maps made by Marco and Elia, a PhD candidate or postdoc, the second one with Stefan, show you actually the samples, if I may say, although I have all the respect to all the participants in this study, which are denoted by a circle. You see in blue the Lake Geneva, and this is the area of Lausanne. These are our participants. And in this blue or red colors on the left, you see the clusters, which are of course clusters that are obtained after rigorous statistical analysis of individuals that constitute a neighborhood which has either a high estimate in the upper left side of their current anxiety level, which nicely coincides also with areas where we have higher air pollution, and the ones who are near to the lakeside, they have less levels of anxiety. And the same on the bottom row left, we see this cluster in the center of the city where we have less green spaces or access to green spaces that show actually a low estimate of their mood. of course, we have corrected and adjusted for many other variables, socioeconomic status being one of the most prominent that comes to our minds. And then, of course, in a more complex triple interaction, we were looking how this link between environmental factors and behavior, be it on the side of affective, of mood, or of cognitive abilities, are also linked to brain anatomy. And here we show these gradients of impact where actually this on the surface of Lausanne environmental factors have a link to our behavior via some differences in brain anatomy. Then, of course, the next big topic for us was temperature change over time. And here I really have to give all the credits to Stefan, who with his younger colleagues estimated actually the land surface temperature in the area of Lausanne in a 20 year span between 1998 on the left-hand side and 2018 to actually here calculate the delta, the difference between the two maps. And we see that there is a certain gradient of temperature changes across the geographical expand of the city of Lausanne. And also here we see certain clusters either with a high or low estimates of mood that are linked to this temperature changes over time. Our next step is, of course, to go to the next level, where we look also at differences in brain anatomy. Well, I judge myself, unfortunately, as being not that savvy in social media, nice ideas as Anna showed. I should be taken care of. But the most imminent impact of our work on climate, thanks to the climate funding, is that we will start the preparation for an ENAC proposal with some German colleagues, and we try to win also colleagues from Norway, where we will be proposing a project on mechanisms, neurobiological mechanisms of resilience and vulnerability to environmental challenges in mental health. The call came just last week, and we prepared already well for this. And in a longer term, for me personally, as a clinician in my career, I will try indeed to make the bridge between what we usually do in medicine, looking at the physical health, but also putting the individual in his or her environment and looking at such effects and interactions. With this, I thank you very much for your attention, and I'm happy to answer questions.
Thank you very much, Bogdan, for this great presentation, very instructive, and for your kind words for the team. It's very appreciated. Now it's my pleasure to welcome Francesco Lazari, who will be representing the project of Marie Violet and Lucas Baumgartner. Francesco got a bachelor and master degree in geological sciences at the University of Padova with a thesis on the frictional power dissipation of earthquakes. Currently, he's doing a PhD on the effect of fluid chemistry on the brittle to ductile transition of rocks and its implication for geothermal energy. His talk is on the effect of fluid composition on the brittle to ductile transition of rocks, implications for deep geothermal energy. Thank you for being with us, Francesco.
Thank you for the introduction. So now I will present to you basically the work I did in the last year of my PhD. We don't have a lot of results yet, but we have some interesting insights and methods that we developed thanks to the CLIMACT grant. So first of all, I will talk a bit about the deformation of geothermal reservoirs, doing a bit of introduction on the topic. Then I will talk about the electrical conductivity of rocks, and I will show you the setups that we developed. And then I'll talk a bit more to conclude about the developments that we aim to do during my PhD. So I will skim a bit on the definition of geothermal energy. I will just say that for those who don't know, geothermal energy is basically the exploitation of a hot fluid that comes out of a rock to generate either heat or electricity via steam turbines with it. A geothermal reservoir is whatever rock basically is capable of hosting a hot fluid that can be exploited to produce geothermal energy. For the sake of this presentation, we will distinguish them in two types. There are porous rocks, that are rocks with a porosity greater than 5%. They have a usually really high permeability, which makes them easier to exploit, meaning that we can just flow water through them and extract it when it heats up in the rock and then generate electricity with the steam. But they're usually not found super deep or in really high temperatures, so that the amount of electricity we can produce is limited. On the other hand, we have tight rocks which have a porosity of less than 3%. They are crystalline rocks, meaning plutonic and metamorphic rocks. They have a way lower permeability, meaning that we cannot flow the fluid through the rocks without generating additional cracks artificially in the rock. But they are found way deeper and in way hotter conditions than porous rocks, meaning that we can potentially generate up to 1 order of magnitude more electricity with the same geothermal power plant. So the idea is to increase as much as possible the electricity that we can produce is to find the hottest rock possible. The only problem with that is that rock change their behavior when we increase the temperature and the pressure. And we might encounter what we call the brittle to ductile transition. So if you look at the graph in the middle, we have a space that is composed of the peak strength of the rock on the Y, so when the rock breaks, and on the x-axis, we have the increase of the pressure and the temperature at which the rock is sitting. On the left part, so at low confinement pressure and temperature, we have the brittle field, in which the strength of the rock is increasing linearly with the pressure and the temperature. So the deeper we go, the stronger the rock is, and the less likely it is to break it, so to have an earthquake. When we go at really high temperature and pressure, we are in the ductile field, which means that the rock doesn't break like we commonly think, but there are more complicated processes that are temperature dependent that come in place. And the rock, instead of breaking, is flowing homogeneously. So we don't have faults, but the rock is deforming like rubber, basically. The transition between these two fields is not sharp. It's an area where the two processes coexist, and it is what we call the brittle to ductile transition. When we look at porous rocks, the brittle to ductile transition is a similar concept, but slightly different. So the brittle field is basically the same. We have the rocks deforming, breaking into faults. In the ductile field, instead, basically porous rocks have a high porosity, so they have a lot of dead volume in them. So when we go at high pressure, when we deform them, they basically crash. They decrease their volume, and so their behavior is a bit different. But still, the transition from brittle to ductile processes is not sharp, it's transitional, and it is what we call the brittle to ductile transition. So in the past, let's say 20, 30 years, there have been a lot of studies on the transition, but there is not yet a complete agreement on what are the phenomenologies of the rocks in this zone. Moreover, in natural geothermal reservoirs are not dry rocks, they are saturated rocks, so they host water. And almost all the times, it's not pure water, but it's water enriched with other chemical species. And the effect of the presence of water itself and of fluid chemistry is mostly unknown on the brittle to ductile transition. So far, we know that in general, just the presence of water weakens potentially considerably the rocks, depending on what rock we're talking about. And there are some studies that show that fluid chemistry, such as the pH or the presence of specific ions, can weaken considerably the rocks. And this, like in a geothermal power plant, means that if we inject the wrong fluid underground, we can cause major earthquakes or have really important subsidence, which if the power plant is close to an urban area, might be a really considerable problem. So we want to avoid this. in order to study what happens to the rocks and to understand how to avoid these kind of issues, we need to replicate in the laboratory the pressure and temperature conditions at which these rocks are, and we need to try to deform them. In order to do so, since we barely know anything about what happens in the rocks with the fluid chemistry, we picked the two relatively easy rocks, which are sandstones, which are basically rocks made of grains of sand welded together. We picked the Fontainebleau sandstone, which is made almost 100% of quartz, which is our non-alterable, let's say, end member that should not react at all with the fluid that is hosting. The second rock that we picked is Adams-Wheeler sandstone, which is made mainly of quartz, but also has K feldspar and around 10% of phyllosilicates that should react with the fluid that we're going to deform it with. To deform the rocks, we use the two apparatuses. The first apparatus is a uniaxial apparatus, which allows us to deform the rock in basically room conditions, so 25 degrees and 1 atmosphere. It's basically when you perform the experiment, the two steel piston press within each other and they break the rock, and we can monitor how the rock breaks, how long it takes to break. The second apparatus is slightly more complicated. It's a triaxial machine, which means that you still break the rock by pushing two pistons against each other, but the rock is also confined by some oil that is pressing on the sides of the rock, simulating the pressure that there is at depth on the rock. We also have a furnace on the outer side of the sample chamber that allows us to heat up the sample up to 200 degrees Celsius. We perform deformation experiments at a constant strain rate, which means that the two pistons of the machine are coming towards each other at the same speed throughout the whole experiment. We kept the pressure of the fluid inside the rock constant to avoid any additional effect of that. We measured the seismic velocity during the experiment, the permeability, every 5 minutes during the experiment, and we sampled the fluid that was coming out of the rock to see how it was changing during the experiment. Now I'm going to show you a bit of the results that we got. In the first panel, you can see the results of the uniaxial experiments, so the experiments with the rocks at room temperature and pressure. On the Y, we have the peak stress, so the stress at which the rock breaks in black. In pale blue, instead, we have the yield stress, which is the stress at which the rock starts to deform non-reversibly. And along the X, we have the different solutions that we saturated the sample with. So we can see that the dry samples are stronger than the wet ones, but between the different solutions, there is no difference at all. So from here, it seemed like the presence of water itself weakens the rocks, but it doesn't matter too much what kind of chemistry the fluid is composed of. On the second panel, instead, we have the experiments under pressure. In black and blue, we have the experiments at room temperature. In red, we have some experiments done at 100 degrees Celsius. Of course, the experimenting temperature are more representative of what's really happening to the rocks. So if you look at the room temperature ones, we barely have any weakening due to the presence of water, and we don't see any difference in the different fluid chemistries. While if we look at the experiment at 100 degrees, we have basically the same strength in the dry sample, but the wet sample with water is quite considerably weaker than the dry one, which means that when we increase the temperature, the presence of water becomes much more important than at room temperature. Here are a few details of the experiments at 100 degrees. Here I'm going to show you a wet and a dry experiment in the first panel done at 20 megapascal of confinement pressure, where the sample was brittle, so it was breaking with a fracture. And on the second panel, there are two experiments done at 50 megapascal of confinement pressure, where the samples were ductile, meaning that they were not breaking, but they were deforming homogeneously. And we can see that in both cases, the black curve here, which is the dry experiment, has a higher stress than the wet curve, which means that the dry sample is stronger than the wet one, both in the brittle and in the ductile field. On the bottom panel, we can also see the permeability variation of the two wet samples. And we can see that with increasing strain, so with increasing the formation of the rock, we are decreasing the permeability of the sample because of closing of pores, mostly. Now I'm going to talk a bit about the electrical conductivity of rocks. First of all, electrical conductivity is one of the main surveying methods that there is to characterize geothermal fields, because it is extremely sensitive to the chemical composition of the fluid and to the presence of clays in the rocks, which are the main proxy to define alteration zones in geothermal fields. And it's probably one of the best proxies for the rock chemistry that we can use. When we measure electrical conductivity with alternating current, it is a complex number. We can represent it as a vector on the real and imaginary plane, and we can describe it with its moduli, the moduli of sigma, which is the conductivity, and its phase. In a saturated rock, so a sample that is composed of minerals and water within the minerals, the bulk conductivity is given by the fluid conductivity, sigma W, divided by the formation factor, plus the surface conductivity of the minerals. So here we can see that the conductivity of a rock is determined by the conductivity of the fluid that is saturating it, which is proportional to the ion content of the fluid, and by the surface conductivity of the minerals, which can roughly approximate it to the content of clays of the rock. In particular, the formation factor, which is what we divide the water conductivity with, is given by the porosity of the rock elevated to -m, which is the cementation factor. So we have a lot of information on the rock by only doing one measure of conductivity. We can also understand the porosity of the rock and how the different grains of the rock are glued together, let's say. This is the setup that we developed in the last year to measure the conductivity inside the deformation machine and during deformation, which is something quite unique that really few people manage to do around the world, we can say. It's a relatively simple setup. So it's a 4-electrode setup, meaning that we impose the current through the sample with 2 electrodes, and we measure the potential difference with other 2 electrodes. In blue, you see the current electrodes. They are 2 copper beryllium alloy plates that are isolated from the machine with 2 zirconium ceramic plates. And the 2 potential electrodes are just 2 stripes of the same metal wrapped around the sample and linked to the instrument and measure the permeability. It's a really simple setup, but it took a long while to calibrate and had it work, because this thing has to resist to pressures up to 2000 atmospheres, so it needs to be really solid. Since it's a new method, we had to benchmark and calibrate it. To do so, we did some hydrostatic experiments, which are experiments in which we put a rock in the machine and we increase the pressure around the rock isotropically. So we don't deform the rock with the 2 pistons, but we just load it with the oil. We did some pressure steps from 1 to 150 megapascals. And at each step, we measured the seismic velocity through the sample, the permeability, the electrical conductivity, and we had a continuous measurement of the volume of the pores during the experiment. Here is one of the experiments that we did. In the first panel, we have the permeability variation with increasing pressure. We see that the permeability decreases a bit, not too much, but that's what we expect, because we are not actually crushing the rock. We are just loading it elastically. In the second panel, we have a measurement, the measurement of electrical conductivity in both 2 and 4 electrode setup, and we can see that both the measurements are quite in agreement with each other. There's a bit of shift between the 2, but can be explained by the resistance of the electrode with the rock. This is something that is well documented in the literature. So with these kind of tests, we demonstrated that we have a method that is working, that is reliable. It's also consistent with the published data that is in the literature, and so we can use it to study electrical conductivity during our experiments. Now I'll talk a bit about what we plan to do with this in the future, because we didn't get a ton of results so far, but we have a lot of ideas on how to use this method now that we proved that it's working and we made it reliable. First of all, we want to keep studying the effect of the presence of water on the brittle to ductile transition. To do so, we will keep doing deformation experiments at different pressures so that we go across the brittle to ductile transition. We'll do the experiments first without water and then with water, so that we can see what's changing between the 2. And during the experiments, we're going to monitor the electrical conductivity, the permeability, the seismic wave velocity, and also the pH of the fluid that is coming out of the sample to see whether some chemical reaction are taking place in the rock. Once we have the deformed samples, we're going to look at the microstructures under the microscope, and we're going to do also some point chemical analysis and some biochemistry to see whether we had any chemical reaction in the rock also after the experiments. After that, we're going to do basically the same, this time adding, instead of pure water, adding chemically more complicated water, which means low and high pH, and we're going to enrich the water in specific ions such as sodium, potassium, and magnesium. Then the rest of the protocol will be just the same. Last, we're going to switch to tight rocks, and we basically what we want to do is to try and understand how the effect of a fully developed alteration, so we will collect already altered samples, affects the brittle to ductile transition. And this is extremely important for really deep geothermal fields that are being developed in Iceland right now, and that allow for a really, really high energy output. And that was it for my presentation. Thank you, everyone.
Thank you very much, Francesco, for this this great talk.